Welcome everyone to today's webinar. We're very happy that you've decided to join us this morning or this afternoon, depending on where you are in the world. As you know, we're here today to talk about how to enhance your research processes through the recently launched integration of Envision Pharma Group and Scite. Let me briefly introduce ourselves before we dive in. My name is Julia Heeson. I'm the Director of Growth Marketing here at Research Solutions, and I'm your moderator today. I'm also here to assist you if you have any questions. I'll be monitoring the live chat, so on Zoom below where it says chat, feel free to reach out to me if you have any technical issues or any questions of that sort. And then also our main presenters and panelists today are Jon Edwards. He's the Senior Product Manager at Envision Pharma Group, and Josh Nicholson, Chief Strategy Officer at Research Solutions and also co-founder of Scite. Welcome to the webinar, Jon and Josh.
Thank you.
A couple of quick housekeeping items today. You're all in listen-in mode, so you don't have to worry about background noise, dogs barking in the background, or your background as such, like the video, everything is turned off. As I mentioned, if you want to reach out, you can use the live chat. I'll be checking that. Then we also have dedicated time for all of your questions towards the end of the webinar. On Zoom below, there's also one little button that says Q&A. So if you click on there, you can add your questions as soon as they come up. So you don't have to wait until the end. You can write them in there as soon as they come up, but know that we'll be answering the questions towards the end of the webinar. And then also this webinar is being recorded, and I will be sending out a copy of the webinar to all of you, so you can rewatch any part if you want to do that, if you want to share it with your colleagues. If you have to drop a couple of minutes earlier, you don't have to worry about any of that. Okay, little overview for the ones of you if you're not familiar with Research Solutions, and then in just a moment about Envision Pharma Group as well. So Research Solutions provide solutions that simplify and streamline how organizations discover, acquire, manage, and then also create scientific articles and data. Today, more than 70% of the top pharmaceutical companies, prestigious universities, and emerging businesses rely on our family of products powered by AI and NLP technologies. Last year, we acquired 2 companies, one of which is called Scite, which you'll learn much more about today. Scite is an award-winning search and discovery platform that leverages AI to increase the discoverability and evaluation of research. I'll leave that up to Josh to talk a little bit more about Scite in just a moment here. Okay, with that being said, Jon, over to you about the brief overview of Envision Pharma Group.
Lovely. Thanks, Julia. So my name's Jon Edwards. I've been part of the product management team at Envision Pharma for a little over 4 years now. So Envision Pharma Group was founded in 2001. We're a leading global technology-enabled strategic solutions partner. We currently partner with over 200 pharma and biotech companies, including all of the top 20 pharma companies. So we support clients across the product lifecycle through a comprehensive suite of service and industry-leading solutions. So we're the #1 provider of medical affairs compliance software solutions, global leader in medical strategy and communications. We currently have scientific and commercial experts across 32 therapeutic areas, and we've recently purchased Okra AI to really sort of enhance our tech and AI capabilities. And over to Josh.
Yeah, thank you both. And I am not in listen-only mode, so apologies if there are any barking dogs, although he's currently asleep. I'm very excited about this partnership, and so I'm going to talk a little bit about kind of the history of Scite, why we started it. I'll talk a little bit about my research as well, and then walk through how, bringing together these products through this integration and this partnership can really be beneficial to all these shared different users amongst the 2 different companies. And so we've been discussing and working on this partnership for quite some time, actually. So Jon and I think, have done various demos and talks together. And so, what is Scite bringing to the table? What is Envision Pharma bringing to the table? And how does bringing the 2 products together really make your life easier, more trustworthy, et cetera? And so to start, I want to talk a little bit about what is Scite? Why did we start Scite? How can this be helpful? What are Smart Citations? To give you some context and some background on that. And then I'll turn it over to Jon at the end to walk through how this integration works directly in Envision Pharma products. And so previously, about 9 years ago at this point, my life was spent making these videos, right? So watching a lot of cells divide, looking at cancer cells, human cells, and looking at chromosome segregation, how this looks. And so this is what researchers are doing around the world. And I was interested in not just my own work at the bench and watching cells divide, but also, research as an enterprise. How do we publish research? How do we fund research? Is it trustworthy? How do we discover it, et cetera? Because everything we do, whether it's watching, cells divide under a microscope, whether it's doing research for policy, all that, is foundational to really kind of the advancement of knowledge. And so towards the end of my PhD, this paper came out. Undoubtedly, many on this call have probably read it. This was written by two former VPs at Amgen, Glenn Begley and Lee Ellis. And what they had written here was really recounting 10 plus years of them doing drug development. And that is, looking at research ideas in the literature, developing a pipeline for that, trying to get it, past clinical trials to make therapies. What they had found, and actually what previous groups had found, is that more often than not, they could not validate major biomedical research findings in the literature. And so this got a lot of attention. It was in Nature, it was from Amgen, and the numbers were pretty alarming, right? 89% of studies, if you can't validate that, that is a lot of wasted opportunity, a lot of money, potentially billions of dollars, and then, of course, people's lives, right? We're developing therapies to help, improve the livelihood of people. And so as an ambitious and maybe naive and idealistic student, I wanted to do something about this. And so back in 2014, again, maybe the last year of my PhD, I wrote an email to the head of the VBI, which is where I was doing my research, suggesting really that we launch a new way to publish, a new way to evaluate research. Again, very idealistic. This email did not get a response, but I think it was kind of ahead of its time. And so here, I am suggesting that we launch something called bioRxiv, based on arXiv, which had existed in physics. And again, bioRxiv is now a thing, right? bioRxiv hasn't solved trust and all these different things, but bioRxiv is used quite a bit. And there's also medRxiv, especially in the pandemic, where there are thousands of papers posted a day, I believe. And so, not to kind of be dissuaded, I entered a business competition. This business competition goes from my email to saying, okay, if I can't convince the head of the department to do anything here, maybe I can just do something myself or with my colleagues. And so again, entered this business competition not knowing anything about business because most of my time was spent watching cells divide, and was rejected, right? The idea that people would post articles online would just be, there would be no need because they could just do this themselves, right? And so again, said, okay, there's some truth to that. I can understand this, but I still think there's something there. And so I started my first company, The Winnower. And the idea behind The Winnower is that we would publish things first and then winnow the good from the bad via open post-publication peer review. The Winnower didn't really collect a lot of research findings, but it did collect gray literature. And that is things that are peripheral to the main research article, such as backstories, such as journal clubs, such as student essays, Reddit science essays, anything that researchers were writing or posting. We wanted to be kind of the central source for that, to give it tools so that it could be cited, so that it could be archived, and so that it could collect metrics. On The Winnower, we published this idea introducing the R factor, a measure of scientific veracity. And so again, this is really in response to those growing concerns around reproducibility that large pharma was starting to put out in biomedical research, and then nonprofits in other disciplines like psychology, et cetera. In this paper, we suggest that, there should be better metrics. We didn't think metrics were going away. We weren't in love with metrics necessarily, but we thought that there was a better way to look at this. And we should not just have metrics around impact, which is currently what we have for the most part, but metrics around veracity, or reproducibility, or replicability. And we almost called it the V factor because I think, sometimes the R can be a bit confusing. In this, we suggest other people build this, right? So we didn't think we could actually do anything like we were writing. So we said Elsevier should build this, Thomson Reuters should build this, reproducibility initiatives should start to look at better metrics around reproducibility. again, ultimately, this is not how things work. Saying someone should build a company or build an idea generally doesn't work that way. You have to do kind of a lot of this yourself. And so the core idea behind this was really trying to improve citations so that you could quickly and easily see if an article had been reproduced or not, right? Incentivize reproduction, track reproduction. Maybe you'll get more of that as opposed to flashy results. Again, as you can see from me sending cold emails, entering business competitions, I was not to be stopped, or we were not to be stopped. My co-founder, Yuri, who is a professor at Cold Spring Harbor, we started to tweet, right? And we said, hey, we have this idea. We've written a paper on it. We've done some manual work here. This is now 2016. So you can see the process from reading about the problem to trying to start to do something by paper to prototype to really build this, right? And so here is me sending a tweet or a DM from a tweet to one of our co-founders that helped us build this early prototype. And so this is a prototype of the R factor, which has essentially become Scite, looking at citations and classifying them as confirming or mentioning. And so as soon as we saw this prototype where you could look up any PubMed ID in this very rudimentary way, we really kind of saw this value, right? Wouldn't it be great if you could look up how any article has been cited to see if it's been confirmed or not? So you could quickly check if an article had been, reproduced or not. And again, to incentivize people to reproduce things so that they would count, right? If you count things, people start to kind of act beyond that. And so we've come a long way to where we are today, to even joining Research Solutions, where we have now, launched a full company. We raised a couple $1 million in grants from the NIH and NSF, and built a product, which I'll walk you through briefly here, that can better help you understand how and why research articles have been cited, researchers have been cited, topics, ideas, etc., and how those smart citations, which we now call them, can even help validate and utilize LLMs in this world, right? Which also has this trust. And you can see, actually, the screenshot's quite out of date because this was only 100,000 scientists, and we're now over 1 million different users. And so what exactly is Scite beyond that prototype? How can that help? And what is, kind of the world as it exists without Scite? And so the way that I generally like to show Scite is to show the world as it exists without Scite. And so here is a research article published in Nature, arguably the best journal in the world, looking at, small molecule inhibitor of USP14. I can't show where these researchers are from, but they're from Harvard. You can see it's been accessed 20,000 times. It has 741 citations, 38 altmetric score, meaning it's been shared widely on social media. This is, by all accounts, a great research article, right? Researchers would kill to have an article like this in Nature to have these metrics. And so the question is this, trustworthy, right? Can we build a drug development program off this? Can we cite this? Do we want to spend our own time and energy, whether that's the microscopy or whether that's something more intensive, building upon this, right? And as Google Scholar has a saying, science, it builds on the shoulders of giants. Really, it is important, right? Because research is very collaborative, not just on single papers, but across time and space. And I think that's, again, really a remarkable thing about citations. It's not just this number and this metric, but it connects ideas across geographies, across disciplines, and again, across time, right? People are still citing research that are hundreds of years old. And so in today's world, maybe you just download the PDF and move on, say, okay, there's a good article, it comes from good researchers, it's highly cited, I can trust it. If you click through to the citations, whether that's Crossref or Scopus or Web of Science or any traditional citation index, what you're going to see is a list of citing articles. No one has the time, or in many cases, if you're a private company, the financial well-beings to open up 600 or 700 different articles. It's just way too time consuming and potentially very costly. And so consequently, no one does that, right? You don't read an article and then read every single citing article to see what it said. But I think that is a pretty big missed opportunity, because behind these 683 citations are maybe 5 years of this person's life studying something very in depth with maybe a lot of grant money, a lot of analysis, a lot of statistics, a lot of data that could be very valuable to how you interpret this article, right? Maybe there's some criticism that they have of the methods. Maybe they utilize the methods and they cite in the methods section. Maybe they are looking at the similar claim and they're looking at a different system so you can build more trust. Maybe they're challenging that. And so there are many different reasons to cite an article, many different ways to cite an article, many different places to cite an article, right? An introduction section citation versus a method section citation are quite different. But in today's world, and historically for decades, really, we have treated citations as if they're all the same, right? And they're not. And we've also boiled them down to a single number where a high number is good and a low number is not so good. And sometimes we have variants of that, like the H index. But again, it's just all counting, right? And so with Scite, we want to go beyond that to make this information more digestible so that you can understand it, so that you, again, can kind of distill the insights from this without spending days or hours, really, which is what it would take to open all these 683 documents. And so if you look at this article through the lens of Scite, or any article through the lens of Scite, what you can see is that we have basically the same metadata as the version of record. So the title, the authors, the abstract. And then what we do differently, and I am expanding this view here, is that we go into the full text of the citing article and extract out these citation statements. And so these citation statements allow you to quickly, easily see what is this conversation happening amongst these papers? How are they citing it? Why are they citing it? And then where are they citing it? And so this first one you can see is extracted from the discussion section. Generally, we're extracting out 3 sentences, and then you can just simply read this, right? And here it's saying the data obtained herein for the GABA B are, however, at variance with this canonical role of USP14 as a proteasomal associated deubiquitinase. It's amazing that I can still say such words, especially after I've been speaking for a bit. And so this allows you to quickly see, okay, here's an article, it was published a few years after it. They're finding some data that's at variance with this. Maybe this is not a direct refutation of this, but maybe this is something that we want to look at. Here is an article published in F1000 Research. This is actually published by the Amgen researchers. And what they're saying is the bottom line of this study is that the manipulation of USP14 level and activity in a cell model system has no discernible effect on the level of these proteins, contrary to expectations based on a previously published study. And so this is a group reporting that they could not reproduce these studies, and then directly reporting that and challenging that. And so that was all there within the citations, accessible for you, but buried, right? Buried amongst 700 different citations. And so this insight, these value can really change, whether you trust that article. It can also help you understand the article, because you can get, maybe a couple or a dozen or hundreds of different single sentence summaries that are written by experts by definition, because they're citing it around the key takeaways of this. And so this is, a really kind of powerful thing that can open up, not just has this been supported or contrasted, but generally how has this article been received in the literature, making citations much more digestible and much more useful than just a metric or just a number. And so to this end of really kind of introducing these new metrics and developing the next generation of citations, we have partnered with publishers to get access to the full text. And so this is it. We've spent significant amount of time, and we're still spending time on this, getting access to the full text of articles. And so we partnered with Wiley, we partnered with American Chemical Society, with Sage. I don't want to say one that's about to come, more and more. And we now have extracted out 1 billion plus citation statements from the full text of the literature. And so we're not just looking at OA content, but we're actually going into the full text of subscription-based content. In addition to this, we now display this content on millions of articles. So every American Chemical Society will display Smart Citations from Scite. Every Royal Society article will display Smart Citations from Scite, American Physiological Society, Sage, Archive, etc. And so this has been a really kind of big trajectory from, hey, here's this email that gets ignored, to now working with leading publishers to really improve smart citations. And as we'll show in a few slides, how this can help, by integrating directly into iEnvision and other data vision or Envision Pharma products. Before going on to that, we are all kind of enveloped in this world of AI and LLMs and how we want to look at that. And so we discussed internally, what does that mean? Can we use LLMs in research? What are the challenges here? And so most of you are well aware that LLMs are extremely easy to use. You don't need to know what a Boolean operator is. You can just give it any prompt you can think of, no matter how wild that prompt is, and it will give you this response. And it will give you a response pretty quickly, pretty confidently, that looks pretty right. But is it right? Is this something that we can rely upon in research to really use? So on the left side, I'm showing a tweet from a researcher who asked ChatGPT to give a bibliography of studies looking at publication bias. It does give a list of studies. They're formatted quite nicely. There's DOIs. JAMA is, of course, a real journal. Some of these are even real authors, but none of these articles actually exist, right? They're a hodgepodge or kind of Frankenstein of all these different things that the LLM is predicting. And so if we can't trust that it can give a real article, how can we really trust any of this input? And so we discussed this internally and, kind of came to the conclusion that what we had been building to help evaluate the veracity of research articles might be quite good for evaluating the veracity of LLM output. And so we launched Scite Assistant. And why I'm showing Scite Assistant is that we're not just citations. We're a very powerful search and discovery system. We do also have a very powerful AI assistant that leverages our unique access to literature to utilize kind of the best of LLMs and chatbots and the best of peer-reviewed articles that, again, have data, undergone peer review, and have, undergone various statistical testing. And so this allows you to ask a question such as, how does the structure of a protein affect its function? It will generate a search strategy. You can see what that search strategy is. You can edit that search strategy. It gives you this nice output. You can hover over these different citations and you can compare. Here's what the machine wrote and here's what we're validating against in the literature. So leveraging smart citations to, again, build trust at the research article level, but then also, with a powerful AI assistant. And so I'm very happy that we're now integrated directly into iEnvision, and as Jon will walk you through, a variety of other tools, because I think this integration just makes so much sense, right? We have this pretty unique indicator, if you will, that allows you to look at how an article's been cited. And in the terms of your guys' use here, this is important, right? It's very critical to see not just how something has been cited, also has it been retracted and things like that. And so I'm going to turn this over to Jon as he can walk us through Scite in iEnvision. And again, happy to answer any questions at the end of this, and hopefully we have some time for that. So
Lovely.
Thanks a lot.
Thanks very much, Josh.
Yep.
Can we have the next slide, please? So very much as Josh has sort of talked about the evolution of Scite, I just really wanted to sort of touch on the industry challenges which led us to develop iEnvision. So you can see that we got the challenges sort of listed out here. So limited enterprise access to plans, no collaborative review of documents, issues around compliance audit risks, no integration with source systems. So this might be your clinical trial management systems, no ability to integrate with them. Tactics that aren't aligned with strategic medical objectives or your SCPs, scientific narratives not managed in real time, and also sort of issues around budgets as well. So all of these things sort of fed in to the development of iEnvision. So, we offer integration of strategy with plans and project delivery, consistency, transparency, and alignment across regions and functions, efficient project delivery, optimized timelines, budget management, reporting and analytics to assess programs and also sort of gap analysis within that, and also full auditability and built-in compliance controls. Can we have the next slide, please? So just to point out that iEnvision is a modular, scalable web-based and secure and purpose-built medical affairs solution. What it really helps you to do is to develop your plans, deliver projects and analyze progress and also the impact of your projects. So what we've done here is we've just set out the various different parts, the modules within iEnvision. So you can see that our scientific communication platform and publication planning tool very much sort of fit in the developing your plans. And then we move across to tools such as DataVision, DoDoc, and Clear, which is where you develop the actual publications. And we also offer a range of research and grants and expanded access, our research and grant solutions as well. Within each of the tools, we have dashboards and reporting capabilities. And just to point out as well that we've just in the last month or so launched our new data warehouse and insights capabilities, which I think will really sort of add to and enhance both iEnvision and our new Envision One platform that we're developing. So library is shown here at the bottom. So that's our sort of our content repository. And you can see on this slide, what I've done is I've just highlighted the individual modules within iEnvision where the Scite integration is available. So it's available within our scientific communications platform, within DataVision, and also within library. And the use cases for each of these tools varies. So typically, sorry, Josh, can we just go back? Thank you. Yeah, so typically a publication manager would utilize the scientific communication platform and also DataVision, but library is often used right across the pharma company. So, you know, everyone within a pharma company could have access to the library. So we thought when we were sort of looking at where it would make most sense for Scite to integrate within our solutions, these were the three that made most sense to us from a business perspective. And in the next few slides, we'll just go through some of the sort of potential use cases and how you might use Scite. So one thing to point out that iEnvision integrate, the integration with Scite relies on the DOI for each publication. So it's for full publications, not for Congress publications. And I've listed out some of the potential use cases for Scite within iEnvision. So I think one of the key ones is to monitor how the medical and scientific community is citing your full publications, and also, as Josh has pointed out, the context of these citations, you know, whether they're just mentioning or whether they're actually supporting your original papers. So that would be available to view within DataVision, Library and SCP. Another important aspect of this would be looking at your competitors' primary, secondary and review papers, and you could do that within Library and SCP, and you may want to see if their papers, their original papers, are being sort of questioned within the scientific and medical literature as well. So that can obviously provide you with some really useful insights. Another thing that you might want to do is within your background publications, so those that are perhaps looking at the incidence and prevalence of the disease areas that you're working in, you may want to compare the Scite information just to help you select the most appropriate background publications to use. And you can also look and see whether any publications have been retracted or erratums, etc., that have been raised regarding those publications. So another use case for you there. Something else that Scite would enable you to do is to look at who's saying what about your work and what they're saying. So this might be something that could be useful to you, perhaps providing medical scientific liaisons with information that they could use in conversations with authors. You know, they may be able to sort of bring up a particular point that's been raised in one of your publications and use that with conversations with your authors and others. Another thing that you could use it for is the assessment of the overall impact of the publication. So using sort of Scite information alongside other measures, you know, perhaps altmetrics, Scopus and things as well, just to give you a kind of a read as to how your publications, what impact they're having. You could also do that versus your competitor publications. And the last thing I wanted to mention, and I think this is quite important, is quite often with large companies, they can license a tool, but people aren't aware that the company actually has a license to that tool. So it can sometimes not be used within an organization. So we feel that the integration with iEnvision, and particularly the solutions that we've chosen for that integration can really help drive the utilization with Scite within your organization. So in the next few slides, what I'm doing is just sort of pointing out exactly where within each of the tools that the integration is available. So within DataVision, the Scite badge is available within the citation node. And you can see it's a little bit fuzzy, I think, on this WebEx, but it appears alongside the altmetric badge and also the Scopus citation count. And one of the things that we've noticed is the number of citations recorded by Scite is pretty much always larger than that by Scopus. And I believe, Josh, I think that that's because you've got a sort of a larger database that you're looking at than Scopus. Is that correct?
Yep.
Super. Next slide, please. Yeah, we also provide the Scite badge here on the Pub Manager's dashboard, and we've got a widget that shows the journal articles that have been published in each calendar quarter. It appears alongside the altmetric badge as well. So the altmetric badge can provide information on whether your paper has been picked up by news outlets and social media, whereas Scite will give you a sort of a more long-term view of exactly how your publication is being viewed in the medical and scientific literature. I mentioned library, so the Scite badge appears both within library records and again alongside the altmetric badge. And next slide. Thank you. It also appears on library portfolio search. So this just provides you with a means of potentially comparing the Scite information alongside each other within the portfolio search itself. Within our scientific communication platform, the Scite badge appears in the horizontal format within the reference list. Just to point out the use case here as well, this is kind of similar to library. So our clients that have SCP typically open up read-only access right across their organization. So this again would be a means of getting this Scite information to many of your colleagues, rather than DataVision, which tends to be sort of limited to those that are actively involved in producing publications. I think I have just one last slide. Yeah, this is just to point out so that some of the information that Josh showed with Scite, all you do is you simply click through the Scite badge, and then that opens up the full integration with Scite. And it's the same for any of the points of integration within iEnvision, be it within DataVision, library, or SCP. You just simply click on it, and then it will take you directly through to the full Scite information. I don't know, Josh, whether you've got anything to add to that.
Yeah, no, I think that's great. Hopefully you can see how bringing Scite into your workflows, can really enhance them. And I think Jon touched a lot on a lot of great points. One is that there are many different tools, and having tools cooperate, I think, has been really big and something that we've seen really helps with adoption and leveraging these things. And so, we don't do what iEnvision different products do and vice versa. And so I think, bringing these together to have really kind of the best strengths makes a lot of sense. On this slide, where we wanted to offer basically a free citation analysis. And so we are, through the product, able to create custom dashboards. These custom dashboards can be on 1,000 articles, on 20,000 articles, etc. Large pharma uses Scite, small biotechs use Scite, universities use Scite, and a lot of them will create these custom dashboards to look at the impact of their own publications. And so if you want to see how having Scite built into iEnvision and other products that Jon just walked through, we're happy to give you a free citation analysis. It's pretty straightforward to do. You just simply need to reach out to your account manager and they can help coordinate this. You can also reach out to Jon, myself, or Julia, and we'll say more. And so again, this is pretty easy to do, and he can help you identify, hey, here's something that we might have missed. And hopefully it's not something like a retracted article that you're unaware of. But again, it's better to be aware of that than not. And so again, reach out to us to this, and we're happy, for you guys joining the webinar to have a free citation analysis from our end. And so with that, I think it's now time for Q&A. And so I'll pass it over to Julia.
Yeah, one quick call out before we dive into the questions. I already see a few. If you like what you saw today, maybe, sometimes after the webinar, you come up with additional questions, or you were like, oh, I want to show this to a few colleagues. I'm adding some links in the chat as I'm speaking right now. So feel free to reach out. I sent one link to Scite, so you can check out the website. Also feel free to reach out, ask for another call, another demo if you want to access a trial, and the same as well for Envision Pharma Group. Or if you're currently a Scite customer and you want to learn more about the integration, vice versa, definitely feel free to reach out there. Also, as Josh mentioned, for the free citation analysis, if you don't know who your account manager is, feel free to reach out to either of us, or also through those same links. You can definitely reach out and just like add as a help topic, like free citation analysis. I don't know who to talk to, and we'll definitely get you to the right person and yeah, get that ready for you. Okay, so diving into the questions here. Also, if you haven't added yours yet, you can feel free to do so. Okay, the first question is, how are pharma companies currently using Scite?
Yeah, so that's a great question. And as mentioned, there's kind of a lot of different use cases, right? Scite can be integrated into, iEnvision products. And so there are groups already using Scite through these workflows that we just have shown. Hopefully there'll be more after this, because again, I think that's a very valuable use case. We also have, large pharma and small biotech using Scite for assistant. A lot of different companies and organizations are trying to figure out their AI strategy and to find ones that are maybe trustworthy that they can be used. And so Scite Assistant is being used across a variety of different companies. That's for internal, communications, but also for marketing, collateral, and things like this. And then, our search is being used alongside traditional searches such as PubMed and things like this that can help you track different products, reagents, diseases. And again, looking at these different dashboards or disease areas, as Jon mentioned, we can help you track not only your own research, your own researchers or affiliated researchers, but also competitors or potentially new ideas. And so really, I think, I liken Scite to glasses. It kind of just helps you see through the literature a lot easier, all this insightful information that can help you make decisions better, make them quicker, and again, avoid potentially some of these costly clinical trials that maybe are not established on solid foundations.
Awesome. Okay, I see one follow-up question here. You mentioned Scite Assistant. Where can I see more? Okay, I'm adding a link here in the chat. Feel free to head over to that link. I think, Josh, correct me if I'm wrong, they have three free searches, right? And then they can see how that works.
Yeah, it's 2. And again, so in terms of the integration, again, reach out to your account manager. It is, as Jon says, like flipping a switch. And so it can be turned on very easily in the tools. We're also happy to meet with you to walk through Scite, in more depth, and we can do it collaboratively. But you can go to scite.ai/assist and run a few queries without even signing in.
Amazing. See a follow-up question on that previous question about how pharma companies are also are using Scite, and the follow-up question is, and how does the integration, how does the iEnvision Pharma Group help with that, Jon?
Sorry, with the integration.
Yeah, how does the integration help companies?
I think it's really just enabling them to quickly access the information. If you want to find one of your own specific publications, you can quickly do that within DataVision or Library just by searching on it, and then you will see the Scite information there. Or your competitor publications, that you may be housing those within Library, and you can just sort of quickly identify those from within that particular tool, and then click through to the Scite integration, and then you'll see all of the information that Josh showed in his slides.
Amazing. Great. Thank you, Jon. Thanks, Josh. Good question so far. Looks like we've got them all answered. So unless there's one last minute question here, I think we're ready to wrap up, and it's always nice to give people some time back in the calendars. As I mentioned, we were recording this webinar, and I will be sending the link. So expect in the next hour or two an email from me. I'll be sending out the recording also with some links if you want to follow up, if any questions do come up afterwards. And yeah, we're happy to take any questions if they do come up. Well, thank you so much, everyone, for joining, spending your time here today. Thank you so much, Josh and Jon, for presenting. And yeah, hope to see you soon.
Awesome. Thank you, everyone.
Bye now.
Bye.